Hi guys, I'm back. My name is Ishul Faisal and welcome to my YouTube channel. So today we will be discussing a very important experiment in biology, and this experiment basically answered the very big and main question which was in scientists' mind during the early 20th century. So that question was none other than this. So basically, DNA or proteins was not the question. The real question was that was genetic information being carried by the DNA or the proteins, right? So was it the proteins that carried it, or was it the DNA that carried it? Now, a lot of scientists were, conducting different experiments. Some were testing on mice by giving them, like different sorts of injections. Some of the injections, one of the scientific experiment was based on the bacteria that caused pneumonia. So there were a lot of experiments, and scientists were there in this race of finding out the answer to whether DNA was the molecule that was containing genetic information, or were there the proteins that were responsible for it. So, in all of that, there was a very clever combination of scientists who worked on the Hershey-Chase experiment. Now, they used the structure of a virus to their advantage, right? So, how does a virus look like? Essentially, they have a genetic material in their in their capsule, and this capsule is basically enclosed by is made up of protein coat, right? So, a genetic material enclosed by a protein coat. So, this is how they look like. So, what were the two essential things that you needed were there in this structure? You have your proteins and you have your DNA as well. Now, I mean, does this like make you think you are reaching somewhere? It does. So, you have got the two things you were differentiating in a single organism. So they decided to use some sort of virus to their advantage, and they shortlisted the T2 bacteriophage. So before we discuss the Hershey-Chase experiment in detail, it's very important to discuss how T2 bacteriophage looks like, what it does, and what's its life cycle. So T2 bacteriophage is basically a virus, and this virus kills bacteria. It consists of a head. Okay. The head contains DNA, and then it has a neck, a collar, a sheath, okay, a base plate, and obviously then some fibers. So what really happens is that this T2 bacteriophage attaches itself to bacteria such as ECOLI. And when it does so, it injects its genetic material into the bacteria. So you can assume that the genetic material goes from here. I'm sorry, it just got erased. Just imagine it's already there, right? So the genetic material goes from here to here. Now, once the genetic material has entered your bacteria, it is going to make it, it's just this external genetic code is going to transform your bacteria by hijacking its synthetic machinery and making copies of itself, and therefore this T2 bacteriophage will be able to make multiple copies of itself inside the E. coli bacteria, the host bacterium, whatever it was. So there will be around 200 of these. Just imagine there are a lot of them. So there will be around 200 of them when the E. coli bacteria would finally burst open, releasing all 200. So this is generally how a life cycle of a T2 bacteriophage looks like. Now that we know how it works, we'll see how the T2 bacteriophage was used for the experiment. Now, scientists, what they did was they tried to answer this wider question of whether DNA was responsible for carrying genetic code or protein codes by, finding out the answer to whether the genetic information being passed on to the bacteria was via DNA or it was via proteins. Now मैंने आपको पहले बता दिया कि it was DNA जिसकी information transfer हुई थी into the bacteria, but scientists didn't know them. So obviously they had to use some sort of marker. And in biology, what really helps out as a marker are radioisotopes. So that is what they exactly did. They took help of two radioisotopes. One of them was sulfur and the other one was phosphorus. Now, where can you find phosphorus? You can find phosphorus in DNA and you can find sulfur in proteins. ठीक है? So, ये अब चीज़ याद रखना, and now I'll take you guys to a diagram that is going to help you understand what happens. Now what they did was they took a huge colony of the T2 bacteriophage and they divided them into two separate columns, and उन दोनों में उन्होंने दो different experiments जो हैं conduct किए. पहली experiment में उन्होंने T2 phage को ना एक phosphorus containing medium में जो है ग्रो किया. अब फॉस्फोरस जो है वो डीएनए में होता है लेकिन प्रोटीन्स में नहीं होता. सो जो आपके डीएनए मॉलिक्यूल्स थे ना, जो T2 बैक्टीरिया के डीएनए मॉलिक्यूल था, उसने जल्दी से वो फॉस्फोरस टेकअप कर लिया, रेडियो आइसोटोप फॉस्फोरस जो कि रेडिएट करेगा जहाँ भी वो जाएगा. और दूसरा जो एक्सपेरिमेंट था उसके अंदर उन्होंने अपने T2 फेज को ग्रो किया इन अ मीडियम दैट कंटेन्स सल्फर. अब सल्फर जो है वो प्रोटीन्स के साथ, ये जो ऑरेंज ऑरेंज हुआ है वो जाके प्रोटीन्स के साथ अटैच हो गया, और फॉस्फोरस आप देख रहे हो पिंक पिंक जाके डीएनए के साथ अटैच हो गया. अब उन्होंने दोनों को जो है ना अपना इकोलाई बैक्टीरिया लिया और दोनों को मिक्स करते हुए उन्होंने एक जो है इनको एक कॉमन मीडियम में डाल के टाइम दिया इसको थोड़ा कि जो T2 फेज है वो बैक्टीरिया को इन्फेक्ट कर ले, और फिर उसको उन्होंने कुछ देर के लिए ब्लेंड किया ताकि वायरसेस बैक्टीरियल सेल से डिटैच हो जाएं. फिर उसके बाद सेंट्रीफ्यूजिंग फोर्सेस जो हैं दे बेसिकली पुट दोज इनटू द सेंट्रीफ्यूज, जो भी उनके मिक्सेस बने उन्होंने सेंट्रीफ्यूज किया उनको. और सेंट्रीफ्यूजिंग फोर्सेस जो होती हैं ना, दे बेसिकली यू नो टुक द बैक्टीरियल सेल्स टू द बॉटम ऑफ द ट्यूब एंड दैट बेसिकली फॉर्म्ड अ पेलेट. सो आपके जो इन्फेक्टेड बैक्टीरियल सेल्स हैं वो पैलेट में चले गए, जो आपके सुपरनेटेंट फ्लूइड है उसके अंदर वायरस हो गया. ठीक है, ये आपकी पैलेट आ गई, ये आपका ये आ गया. ठीक है, ये आपका आपके सुपरनेटेंट फ्लूइड में क्या होगा ये बात याद रखना, जो ऊपर रह जाता है वो आपका सुपरनेटेंट होता है, जो नीचे आ जाती है पैलेट सॉलिडिफाइड वो आपकी पैलेट होती है. सो आपकी जो पैलेट है वो डीएनए से बनी हुई होती है क्योंकि डीएनए ज़्यादा हैवी होएगा. ठीक है, सो आपकी पैलेट जो है उसको हमने किया था कलर विथ फॉस्फोरस. ठीक है, अब नीचे जो है फॉस्फोरस की पैलेट का पिंक कलर आ रहा है और ऊपर क्लियर कलर आ रहा है जो बायोमार्कर से मार्क्ड नहीं थी. तो इसका मतलब कि डीएनए एब्जॉर्ब हुआ है. ठीक है, बैक्टीरिया के अंदर डीएनए गया है. और इस केस में आप देखो जब उन्होंने सल्फर से मार्क किया, सल्फर वाज इन द वायरसेस के प्रोटीन कोड्स ऊपर रह गए थे जो है सुपरनेटेंट फ्लूइड में, और बैक्टीरिया ने नीचे रह जाना था. बैक्टीरिया क्लियर आया है. जब वो क्लियर आया है तो इसका मतलब है कि बैक्टीरिया के अंदर प्रोटीन कोड नहीं गया. बैक्टीरिया जब पिंक आया है तो इसका मतलब है बैक्टीरिया के अंदर डीएनए गया है. जो ऑरेंज मार्कर था जो सिर्फ सिग्निफाई कर रहा था उनको आपके प्रोटीन को, वो ऊपर रह गया है. अगर ये नीचे ऑरेंज होता तो फिर हम कह सकते थे बैक्टीरिया में प्रोटीन कोड गए हैं, बट वो ऑरेंज नहीं है, वो क्लियर है. जो एक्सपेरिमेंट हुआ इसमें वो क्लियर है और ऊपर जो है वो ऑरेंज है. जबकि एक्सपेरिमेंट 1 में जब डीएनए जो है पिंक मार्क्ड है विद द हेल्प ऑफ जो है फॉस्फोरस, तो आपके पास पिंक नीचे आ रहा है, विच मीन्स डीएनए गॉट ट्रांसफर्ड इनटू योर बैक्टीरिया. So this is it about Hershey-Chase experiment, which basically demonstrated that the T2 DNA contains information which is required to make more copies of T2. Okay, so you can explain see the process जो मैंने आपको पहले बताया था about the life cycle of a bacteriophage T2, which attaches itself to the ECOLI bacteria and it transfers its genetic material into it, and once it has transferred it, वो 200 copies उससे burst होके निकलती हैं. So that's it. I hope you guys understood the concept, and if there are any questions you guys can ask them from me.